I'm a senior at St. High School, and my presentation is about biomarkers in lung adenocarcinoma. What is lung adenocarcinoma? Lung adenocarcinoma is a type of lung cancer, and lung cancer is defined as excessive and continuous cell growth resulting in the formation of tumors in the lungs. Biomarkers are an important part of cancer because, and some examples of them, of the molecules are like DNA, RNA, and protein, and they indicate normal processes taking place in the body. Their important role in cancer is in diagnosis and treatment. The objective of my research project was to identify potential biomarkers, candidate hub genes, and key pathways to help improve upon prevention, detection, and treatment of lung adenocarcinoma by better understanding the dysregulation of microRNA and the effects it has in lung adenocarcinoma. The first thing I did was I used the Gene Expression Omnibus database, which is a publicly available database, and I used 7 different data sets from the database, and these are all genome-wide association studies that compared the expression of microRNA in tumor cells and non-tumor cells of lung adenocarcinoma patients and normal patients. In Cytoscape, I identified hub genes, and these hub genes were the important nodes that had many interactions with the partners. With an upregulation in the microRNA, that would lead to a downregulation in the target genes affected with that. And these target genes are the ones that I identify, and therefore it shows that downregulation in the target genes is indicative of a worsened or severe case of lung adenocarcinoma. So the summary and conclusion of of my research project, I identified 5 signature microRNA in lung adenocarcinoma through an in silico analysis using Geo database and Geo2R. Then I discovered 511 genes to be common targets of the differentially expressed microRNA in lung adenocarcinoma. Then from these 511 genes, I constructed a PPI network and identified 10 hub genes that were differentially expressed in that network.
Really nice talk. I want to congratulate you and your mentor, Dr. Syed, for accomplishing such great work. I had a basic question. So in your in silico analysis, what was the reason behind narrowing down to the microRNA, the genes that were targeted by all 5 microRNAs instead of maybe microRNAs that target a single gene or 2 genes or any other combination?
It just showed that the genes that were impacted by all 5 microRNA was more significantly involved with lung adenocarcinoma, and therefore I could focus more on those.
And I'd like to thank you all for attending this meeting, this research project. So my project was on identifying key candidate biomarkers in muscle-invasive bladder cancer using in silico analysis. So what is bladder cancer? Bladder cancer is the proliferation of urothelial cells in the bladder, and this is also known as urothelial carcinoma. So right here on the right, you can see a structure of the bladder. It's made up of, it's connected to the ureters, the bladder, and the urethra, and it expands to accommodate urine like a balloon. Factors like the family history of having bladder cancer, as well as different types of biomarkers, which I know Arjun mentioned before, could also be significant towards the risk of bladder cancer. Well, this research is significant because the prognosis, although it's improved slowly, it's not to the extent where bladder cancer can be mostly non-fatal. So here's my objective and my workflow. I have a chart here to show my workflow. I began with acquiring muscle-invasive cancer, bladder cancer data sets from the Gene Expression Omnibus database, which is GEO. For acquiring the normal bladder and muscle-invasive data sets, like I mentioned before, I used GEO database, which is an online tool to, as an online database, for genomics input by researchers. So here I conducted a gene ontology functional analysis after that. This is the chart for upregulated genes. CDC20 was really the only hub gene out of the 10 that qualified as a prognostic biomarker for muscle-invasive bladder cancer. So as a summary and conclusion, my differentially expressed genes showed that 217 were commonly upregulated, while 334 were commonly downregulated from both datasets acquired from GEO. Then the gene ontology showed us that these overrepresented biological processes, which all of which had some degree of association with carcinogenesis, as you can see here.
So that was a wonderful talk, and again, congratulations, and Dr. Sayed. So I have a couple of queries. So Chris, when you were talking about normal samples, do you know like whether these were normal areas from the patients or these were tissues from healthy individuals?
They were tissues from healthy individuals.
Okay, thanks. And then in the cell cycle upregulation, do you have information about the individual stages of cell cycle, like G0, G1, S phase or something?
Yeah. So basically, some of the genes regulated the G2 or M checkpoint, and that's a major checkpoint in DNA replication, I believe.
Okay.
Okay. Good afternoon. Thank you all for coming. I'll be presenting my work on the identification of mutations in rifabutin-resistant Acinetobacter baumannii, or A. baumannii for short. So antibiotics work by inhibiting different processes in the bacteria shown here. For example, like penicillin prevents the synthesis of the cell wall, whereas clindamycin binds to the 50S subunit of the ribosome and disrupts protein synthesis. Antibiotic resistance occurs when a bacteria has been exposed to an antibiotic multiple times, and it mutates itself in order to become resistant to the antibiotic. And this resistance develops over a long period of time. So A. baumannii is classified by the World Health Organization as a top priority for a need for a new antibiotic, and by the CDC as one of the most dangerous bacteria. Rifabutin is the most effective against A. baumannii, and in this graph, we see that it takes a much lower concentration of rifabutin to kill 50% of an A. baumannii sample compared to all the other antibiotics in that family. And in this study, the researchers also cultured HEMC1 with rifabutin, from which 22 resistant colonies were isolated and then sequenced. And these 22 genomic sequences, along with HEMC1, comprised of the data set that I used in this project. So based on the previous study from USC and the availability of the genomic DNA sequences, the purpose of this project is to compare the genome of a rifabutin-resistant strain of A. baumannii to the genome of a rifabutin-sensitive A. baumannii, or HEMC1, to identify mutations in genes with relevance to rifabutin resistance. And the purpose of trimming these sequences was so that there could be a maximum alignment between the reads and the reference genome, as sequencing adapters are not part of the genome. And after trimming, I checked the quality of the reads using a Galaxy tool called FastQC. Looking at the mutations in 16 or more genomes, I found one in the gene MURI. MURI is responsible for catalyzing the peptidoglycan layer during cell wall biosynthesis, and due to this being an important function, it is a strong target for antimicrobial action. In conclusion, over 200 mutations were found in 16 or more of the genomes, and mutations were also identified in several protein families with potential involvement in antibiotic resistance, such as cell wall biosynthesis, transcriptional regulators, and efflux proteins.
If you have to analyze, like, for example, 50 genomes at a time.
Lab equipment, I would actually try to see if the genes that I described actually affects resistance. So, yeah.
I'm going to be presenting on the antibiotic resistance of Acetara baumannii, or A. baumannii for short. Let's begin. So what is A. baumannii? A. baumannii is a bacterial pathogen, or a bacteria that causes diseases. It is most commonly found in hospital settings, and it is known to cause infections and illnesses like pneumonia. These 22 samples, along with the HMC genome, make up the dataset that I used in my research. So the goal of my research was to compare the HMC1 genome to the 22 rifabutin-resistant A. baumannii samples. With this comparison, we would be able to identify specific DNA mutations that help us explain the antibiotic resistance of A. baumannii. So this is a quick overview of the methodology of my research. After trimming, I ran another tool called FastQC, which allowed me to assess the quality of my reads. After filtering over 45,000 mutations, we were able to find 200 mutations that occurred in 16 plus of our 22 samples. Focusing specifically on one mutation at this position, we can see that the reference sequence reads a G while the samples read A. Finally, as I conclude my presentation, I'd like to restate some of my results. I found over 200 mutations that occurred in 16 of our 22 DNA samples, and through the analysis of these mutations, I was able to form a testable hypothesis that could explain one form of A. baumannii antibiotic resistance. I learned much more than I could have imagined, such as the entire process of DNA sequencing and how to use Galaxy and its wide array of tools. And my understanding of the bioinformatic world is miles ahead of where I started this summer.
You know if there are trials or people are using like multiple antibiotics to cure diseases caused by this bacteria?
So that's a little bit outside of the scope of the research. Based on like the USC study, they were trying to find one antibiotic that could like do it all in a sense. And so once they identified rifabutin as that one antibiotic that could do it all, they also were able, in their trials where they're testing for potential resistance, they identified 22 rifabutin resistant samples, and their methods weren't as transparent as we wanted them to be. So we took those 22 samples and performed analysis to understand like why it was resistant to rifabutin. So that was a little bit out of the scope of the presentation, but in my future research, I'll surely look into them. So thank you for that.
Okay, thank you.
Out of curiosity, so what's the coolest thing you experienced when you access the data with the Galaxy?
Yeah, so definitely one thing that I was very new to the bioinformatic world before this entire research over the past few months. And so one thing was just like one thing that really surprised me was just like the ability to take patterns and use computational analysis to make, to like push forward accurate theories. And it was like there was a lot of having like a biologist hat or like a computer science hat, and just like combining those two worlds was really interesting. And so that was just something that was really surprising and really intriguing on how you can use those two things in tandem to like make discoveries like this. So yeah.
Good. That's nice one.
Step 6. This step was like a manual process, and I'm planning to write a program that's going to allow me to automate this using Python. So that's just like one direction of further research that I'd go. And then obviously, if I was given access to a lab, I'd be able to test more. I'd be able to look into more about the changes of the ABQ protein. So those are just two routes I'd go for future research. So, yeah.
Excellent. Yeah.